Hello, my name is Randall King. I will be presenting on antibiotic resistance of fecal Escherichia coli in poultry, poultry farmers, and poultry slaughterers. Escherichia is just another word for E. coli. This is pretty much my presentation outline. This just gives the gist of how the presentation is going to flow so you can follow along with it. So a little bit about the introduction. E. coli is a gram-negative rod-shaped bacteria, and generally this bacteria is going to be found within the intestines of poultry. In addition to poultry, it can also be found in humans as well. There are strains of E. coli that does have the ability to actually induce diarrhea, and diarrhea is one of the major causes of poultry death because diarrhea doesn't only just spread the bacteria of E. coli. In addition to that, it also causes dehydration in the poultry, and that's one of the main contributors in which the poultry dies due to dehydration through excess feces loss. So this is a little bit about the introduction and continuation of. Salmonella and E. coli are two of the biggest pathogens that are known, especially to the poultry industry as it relates to food products, and it does have a major impact on public health. And that's one of the concerns that we're going to be trying to focus on and try to find a way to mitigate that risk to the poultries, and not only the poultry, but as well as the public health, because the general public are those that are consuming these poultry products, whether it's meat or eggs. So a little bit about the poultry continued. So there have been studies that show there has been an elevated resistance in the E. coli bacteria due to heavily antibiotic drugs being administered. Now, in a sense, it's like a superbug. So the more antibiotics that are given, the bacteria began to develop a high resistance to it. So E. coli spreads not only through feces, but it also spreads during egg development as well. And that's going to be one of the major things that we need to pay attention to. So the infundibulum, that's going to be one of the major important parts that we need to kind of focus on. So the infundibulum is sort of like the fallopian tube in mammals, and that connects to the magnum and the isthmus. So a little bit about that in detail. So the reproductive tract of poultry is divided into two major parts: the ovary and the oviduct. The ovary is pretty much where the yolk is going to be added, and once that yolk reaches an ideal size, it's going to be released into from the ovaries in a process that's known as ovulation. So this yolk is going to be released in the infundibulum, and that's where fertilization occurs, and that's one of the primary locations that the bacteria, the E. coli, is going to attack. So the yolk passes into the magnum, and the magnum is going to be responsible for actually adding the albumin into the egg. Now, once the yolk leaves the magnum, it's going to continue on, and it's going to go into the isthmus. That's where additional portions of the egg are added for the formation of the shell structure. Now, from that portion, so from the isthmus, it's going to travel down to the shell gland. The shell gland is the equivalent of the uterus, and that's where the egg gets its pigments from. So the transmission, the spreading of E. coli, it is known that E. coli is transmitted from the poultry. Of course, it's going to be correlated to human transmission as well, because again, the E. coli is found within the poultry. We as consumers consume the poultry products, which is going to be the meat. So a large portion of the farms that the research focuses on are located within the southeastern region of the Netherlands. And although there are a lot of poultry farms, this doesn't only include chickens in regards to poultry. So there are also turkeys, broilers, which are meat birds, as well as hens that are egg producers. And the primary overall thing that it's going to be focused on is mitigating the outspread or spread of E. coli. That's going to be one of the main focuses at this point. In regards to the materials and methods for the research, fecal samples had to be collected, and those fecal samples were collected from both the broilers as well as the egg-producing hens. In addition to this, questionnaires were actually given to the farmers, and these questionnaires pretty much entailed how much or what dosage of antibiotics the farmers were giving to the poultry. In addition to that, additional information was gathered from the farmers in order to identify if in fact other animals had actually been kept on the same property near the poultry themselves. In regards to the materials and methods, as far as the experimental procedure is concerned, there were actually 11 samples of E. coli, and there were 11 different samples of antibiotics that were administered to those agar plates that contained that bacteria, that E. coli. And this was done in order to see if there is any resistance in regards to administering these antibiotics to the agar plates filled with the E. coli. When testing the fecal samples, it was completely done at random, so there was no exact order in which those samples of feces were actually tested with those antibiotics. In addition to that, they were paired with different antimicrobial fighting agents. Again, those fighting agents are going to be those different antibiotics. This chart displays some of the results that was retrieved from the actual study itself, and this chart simply illustrates the number in which the E. coli strain became resistant in different poultry. So that's in regards to the turkeys, the broilers, as well as the laying hens. And again, this chart simply shows the resistance that was found in the feces of each of the different poultry. So based on the results, some of the main findings actually provided evidence that the common transmission is in fact brought on from E. coli, and this common transmission goes from poultry to human, and this transmission is spread about through the consumption of poultry products, and those poultry products include the eggs as well as the meat of the poultry themselves. It is, however, also important to take into consideration that generally on the average, there are low percentages of humans actually contracting E. coli from consuming poultry products. However, it does occur. So from looking at the research, there is a clear determination that there is in fact a high or moderate amount of resistance of the E. coli due to onset or common administration of antibiotics. So another and an alternate method does need to be discovered in order to reduce the onset of E. coli in poultry.